So, welcome to Biomindscape. Today we are going to know how to write a genetic code. So, the dictionary your cell used to convert genetic information into protein and the building block of life. So, let's begin. So, the genetic code, it is a set of rules by which the information in nucleotide sequence of a DNA or RNA is translated into sequence of amino acid in a protein. So, it is universal and almost all organisms use the same code. So, it is a triplet base, even three nucleotides that are known as the codon and a code for one amino acid. If your mRNA has a sequence of AUG, it will code for methionine, which also acts as a start codon. Now DNA versus RNA, nucleotides. Let's reverse the nucleotide bases. For example, DNA consists adenine, thymine, guanine and cytosine. So what we will write? ATGC. And RNA consists adenine, uracil, guanine and cytosine. So instead of thymine, we have uracil here in RNA. So we will write it AUGC. So in RNA, uracil will be replaced by the will be replacing thymine. So here we will use uracil in RNA and thymine in DNA. So let's know the steps how to write it. So we have taken a DNA from 3' to 5', TAC, CT, AAA, ATT. So this is what we are going to transcribe to mRNA. Now let's make a complementary of 5' to 3'. Now wherever there will be a T, we will write it A, and where instead of T we will add U wherever there is complementary. For example, A complementary, for A the complementary is T for a DNA sequence, whereas it will be U in RNA. Here we are using RNA, so wherever we are using T, we will use U. Instead of T, we will use uracil. So let's write it. So break into the codon. First one is the starting codon, we get AUG. Then the second one we get GGA. Then UUU and UAA. So in such a way we have broken the codon and use the codon table for translating. So AUG will represent the amino acid methionine, which is the start codon. Then GGA will represent the glycine. Then UUU will represent the phenylalanine and UAA will represent a stop codon. So final protein sequence we get is methyl, then lysine, then phenylalanine. So the starting codon methionine, which helps in the initiation of the protein synthesis. Then stop codon, it is UAA, UAG and UGA. They are the terminator for the protein synthesis. So triplet codon, each codon has a 3 nucleotide, non-overlapping. Codes are read one after the another and it is universal. Same codon in almost all the organism, that is a universal codon. And degenerated is the one in which multiple codon can code for a same amino acid. For example, UUU, UUC both code for a phenylalanine. Unambiguous codon, each codon codes for only one amino acid. Whenever you are writing a genetic code, always remember that the codon must be in 5' to 3' direction. Use the mRNA codon table, not DNA, when translating to amino acid, and don't forget the start codon, that is AUG, and the stop codon, which can be UAA, UAG and UGA. Now what are the other names for UAA, UAG and UGA? Write in the comment section. Then let's quickly see what we have learned till now. DNA template, which is from 3' to 5'. For example, TAC, so the mRNA will be from 5' to 3', which is complementary. Wherever there is U, wherever there is T, it will be replaced by U. So we will, in mRNA it will be AUG. And amino acid, which for this it is a methionine. So this is how a simple codon become the first step toward building the life. So now let's know the table. How to draw the table? First we have taken UAGC sequence in this horizontally as well as vertically, and now we are going to make the table. So first we will add here UU, then U. UAGC will be vertically written and UU will be common in 4 times. Now you can see here. Similarly for other codons also we have to make it. So first we have to understand the structure. First position, first base of the codon is shown vertically on the left. Second position, second base of the codon is shown horizontally across the top. And third position, that is the third base of codon, is shown vertically on the right. So each cell is grind represent one codon, and inside the cell you will find the 3-letter codon. So amino acid like methionine, glycine and serine, some cell says stop, and these are stop codon. So let's draw. Make a big 4 into 4 square which consists of 16 cell inside, representing the combination of first and second base. On the left side write the 4 RNA base, that is UCAG downward, and this is for the first base. Now top right write UCAG across, and this is for the second base. Now fill the 3 base and codon information. Each big cell now represent a unique combination of first and second base. Within each of these 16 boxes there are 4 codon with base on the third base is very common, that is UCAG. This one is common in the third base in all of these. You can see the table. So write 4 codon changing the third base, example UUU, UUC, UUA and UUG. So next to each codon write the corresponding amino acid, use the chart you provide. So in this way we can write it, where UUU is a phenyline, and UUA and UUG is the leucine. So do this for all the 16 blocks using the codon chart provided. Always use RNA base, that is AUGC, and not DNA, that is there will be no T. So highlight the important code, starting codon, that is a methionine, and the stop codon, that is a terminator translator, and that is we can represent it by UAA, UAG and UGA. These are the stop codon, and AUG is the start codon. So you can use color to code the amino acid if drawing by hand for memorization. So AUG is always a start codon and stop codon is A or G, that is AUG or AUAG. So end will be having A or G for this stop codon. It is a easy way we can say that UCAG is repeated in all the third type, that is the third codon is UCAG in all of this boxes. You can see here UU, UUC, UUA, UUG. Similarly UCU, UCUC, UCA, UCG. Similarly UA, U. Then UAC, then UAA and UAG. In this way we can write all of these. Now I am marking it with color so that you can see the repeated one UCAG in all of these. Thanks for watching. Do like, share and subscribe.